Hello guys, and welcome back to another video on my YouTube channel. In today's video, I'll be talking about a project that I did for the past 4 to 5 years that was recently accepted into a journal that is soon to be published. So with that, let's get started.
And the title for this year's project is Mitotic Inhibitors Suppress the Tumor Formation and Metastasis of Triple-Negative Breast Cancer. Recently, many papers have shown the high expression of CDC20 in various cancers such as lung, pancreatic, and prostate. However, its expression in relation to breast cancer wasn't clear. Therefore, using a database from NCBI, we were able to find the high expression of CDC20 gene in 3 triple-negative breast cancer cell lines only. Furthermore, the high expression of CDC20 was correlated to a worse overall distant metastasis-free and relapse-free survivals of breast cancer patients. We then confirmed this data through in vitro experiments. Interestingly, we were able to find that mitotic inhibitors, including a CDC20 inhibitor, were also successful in the inhibition of TNBC cell growth and invasion, therefore suggesting the use of CDC20 as a potential marker and therapeutic target, and mitotic inhibitors as a potential additional therapy for triple-negative breast cancer. Breast cancer can be characterized into their respective subtypes based on their expressions in the estrogen receptor and/or progesterone receptor, or the overexpression of the HER2. Among the different subtypes, triple-negative breast cancer is the most aggressive and has the worst prognosis, meaning that they can easily metastasize and has the worst survival rate among the different subtypes. CDC20 or cell division cycle 20 is an oncogene, and it is also a key mitosis regulator. Several substrates of CDC20 or CDH1 and CDC20 itself has been shown to be highly expressed in several human cancers.
So to step in here for a little bit, I want to give a more simpler definition of what CDC20 is. So CDC20 is this gene that is very important and vital in the cellular process called mitosis. And mitosis is the process when cells divide into two, as shown in the picture over here. And because I mentioned in the presentation itself that CDC20 is known to be an oncogene or an oncoprotein, and basically what this means is that CDC20 promotes the characteristics of a cancer within the cell compared to blocking the cancer characteristics. And furthermore, I mentioned the word substrates here and gave several other genes as an example. And what I mean by the substrates is that the CDC20 gene controls other genes as well within the mitosis process, and the substrates stood for the other genes that CDC20 controls.
Here is the schematic of how I downloaded a 53,805 gene dataset for a gene expression analysis through the NCBI GEO website.
Among the 53,805 genes that I downloaded, the database that I downloaded itself showed an expression of whether these genes were expressed in either the hormonal positive breast cancers or the triple negative breast cancers, and I specifically focused on the genes that were highly expressed in only the triple negative breast cancers in this particular project.
So here are my results. Here I found that among the different mitosis-related genes, CDC20 was the only gene to be highly expressed in all 3 triple-negative breast cancer cell lines and not expressed in all 3 hormonal positive breast cancer cell lines. Then using the Breast Cancer Gene Expression Miner version 4.5 with 4,712 human breast cancer patients, I found that the CDC20 gene expressions were highly conserved in the triple-negative breast cancer subtype compared to the other subtypes of breast cancer. Furthermore, the high expressions of CDC20 was associated to a worse overall distant metastasis-free and relapse-free survival of the breast cancer patients.
And here I would like to personally thank the 4,712 breast cancer patients that gave their consent to use their data and information through a public data set. Although I know that that was probably not the easiest choice to make, I would sincerely like to thank the 4,712 breast cancer patients that did give their consent.
Next are the in vitro experiments. We were able to confirm through the Western blot analysis that the CDC20 was highly expressed in only triple-negative breast cancer cell lines. And furthermore, the knockdown of the CDC20 gene in triple-negative breast cancer significantly decreased its breast cancer cell growth, migration, and invasion rate through the colony-forming, wound healing, and Boyden-Chamber assays.
So I would like to jump in a little bit here and explain what and why I was doing these experiments. So the first experiment that was conducted was the Western blot experiment. And what this Western blot experiment does is basically seeing if a certain protein is expressed within a certain cell line. And in my particular project, I was trying to see if the CDC20 proteins were actually highly conserved within the triple-negative breast cancer cell lines, because previously through database results, I found that the CDC20 gene was highly conserved in patients, especially in the triple-negative breast cancer patients. The second experiment that was conducted was the colony forming assay. And basically what the colony forming assay does is it checks the cancer cell growth. And in my experiment, I was trying to see if the knockdown of CDC20 or getting rid of the CDC20 expression within the cell line itself for a temporary amount of time would actually decrease the breast cancer cell growth. The third experiment that was completed was the wound healing assay, and the wound healing checks the cancer cell migration. And in my experiment, I was trying to see again if the knockdown of CDC20 would decrease the triple-negative breast cancer cell migration. And finally, the fourth and final experiment that was conducted in this particular slide was the Boyden-Chamber assay. And what the Boyden-Chamber assay does is it checks to see the cancer cell invasion/infiltration levels. And through this, again, I was trying to see if the knockdown of CDC20 would decrease the triple-negative breast cancer cell infiltration/invasion. And specifically the migration and invasion were a little bit important because triple-negative breast cancer, as I mentioned previously, is known to be one of the most aggressive types of breast cancer, which means that they easily move to other organs, and that's basically like infiltration and invasion and migration.
Next is a highlight of my results. We used APSIN, a CDC20 inhibitor, ZM447439, an Aurora inhibitor, and a BI2539, which is a PLK inhibitor, and checked to see their effects on TNBC cell growth, and found that they significantly decreased the TNBC cell growth. Furthermore, we tested the mitotic inhibitors on TNBC cell migration and invasion and found that they once again significantly decreased the migration and invasion rates.
So what basically happened for the past two figures were that I basically did the same experiments that I previously mentioned, except I did the colony forming, which checked the breast cancer cell growth, and the wound healing or the breast cancer cell migration. And except what was different this time was that instead of temporarily removing the CDC20 expression within the triple-negative breast cancer cell lines, I actually use mitotic inhibitors. And as I kind of explained in my presentation, these mitotic inhibitors all target different proteins that are essential for the mitosis process. And the specific inhibitors that were in use were a CDC20 inhibitor, which was like my main gene protein that was studied in this particular project, and then Aurora, and then PLK.
So in conclusion, I was able to find that CDC20 is highly expressed in triple-negative breast cancer human samples, and that second, the high expression of CDC20 is associated to worse clinical outcomes, that CDC20 is highly expressed in triple-negative breast cancer cell lines, and finally, that mitotic inhibitors could become a target therapy for triple-negative breast cancer and prevent the TNBC cell migration and invasion.
So thank you so much for watching today's presentation on my project that I've been doing for the past 4 to 5 years. And I'm honestly very excited to see my work finally getting published into like the public world. so that not only my viewers can see it, but just anyone and everyone around the world can actually look at my findings and results and study and either be inspired or given a little hope or given inspiration by it. And one thing I would like to address again is that my focus for starting this YouTube channel was first, of course, to inform everyone that is watching this video on breast cancer and just update everyone on how breast cancer is, what are good ways to treat or think about it in a patient/family's perspective, or the different results in different papers that I'm reading and all that, but also to find potentially other teenagers and other teenage researchers that are particularly interested in or just in cancer research in general, specifically breast cancer. So I really hope that if there is someone out there that is around the teenage age that is actually interested, please leave a comment down below or share with someone that you know that is actually interested with the breast cancer research and that are teenagers, so that hopefully one day we can actually meet and we can celebrate what we've learned and shared with the world about breast cancer and what we've done in general about breast cancer and its improvement. Because many scientists and even myself studying for science fair and my projects in general, I feel that we all have this goal of the cure of cancer, and I believe that these small projects, small steps at a time, could in the future later build up to a actual cure. And I've read a book and I saw somewhere that this particular scientist said that you live longer in order to live longer. So basically saying that there's going to be more innovations and drugs out the longer we live, so that we live longer. And I believe that's a very true saying, because we want to develop a cure for cancer and we're trying to find that particular answer to our question. So thank you for watching, and again, please leave a comment down below if you know someone that is interested, or if you are interested, and share with others if you know someone that is particularly interested with the breast cancer research. Thank you again.